Well, welcome everyone back after the lunch break for the session number 3 towards clinical application. My name is Magdalena Riedl Khursigara. I'm a physician scientist and a postdoc in Anna Grecka's lab. So this session is very near and dear to my heart. So let me introduce you the first speaker. His name is Dr. Holt Sakai, and he's a postdoctoral fellow in chemistry and chemical biology at Harvard Medical School in the laboratory of Dr. David Liu. He completed his PhD at Princeton in the Macmillan lab, and in the Liu lab he works on prime editing approaches, which he's going to tell us all about today in his talk, In Vivo Prime Editing Rescues Alternating Hemiplegia of Childhood in Mice.
Okay, nice stand here. Okay, thank you for the introduction. Today I'm excited to share with you some unpublished work on prime editing to rescue a rare neurological condition called alternating hemiplegia of childhood. And so this is a rare disease, and I hope that some of the meta lessons that we're able to extract from this work could be applicable to your rare monogenic disease of interest. And so alternating hemiplegia of childhood is a debilitating neurological condition where patients suffer from a number of symptoms, including paralysis, typically on one side of their body. They also have a number of other symptoms, including involuntary dystonic contractions, and generally can suffer from poor coordination as well. Symptoms appear within the first few months after birth and can lead to developmental delays, and patients are ultimately, in many cases, unable to live independently. So mutations in the ATP1A3 gene cause AHC, and specifically in the alpha-3 subunit of the catalytic trimer. Mutations occur throughout the protein coding sequence of ATP1A3, but primarily cluster within the central transmembrane region. Mutations are heterozygous, de novo, and usually autosomal dominant. And quite interestingly, the vast majority of AHC cases, in fact, 2/3 of them, can be accounted for by just these 3 mutations alone: aspartate 801, glycine 947, and glutamate 815. So for patients with AHC, there are currently no disease-modifying treatments available. They typically rely on symptomatic care. They are highly dependent on caregivers, given that at any moment they can suffer one of these debilitating attacks. And in some cases, in the most severe cases, patients can regress and really is a quite serious disease. So to address the underlying therapy, researchers at Duke developed a mouse model of the disease that recapitulates some of the key hallmarks of this disease. And when they deliver into this mouse model the humanized ATP1A3 transgene under control of a neuron-specific promoter, while they do observe some benefit according to certain metrics, they ultimately are unable to find any durable rescue out to 70 days of these clinically relevant phenotypes. One reason why transgene complementation might have been unsuccessful in this case is that actually in vitro studies in xenopus oocytes have shown that relative to wild-type healthy alleles, inclusion of dominant negative human alleles exert this dominant negative effect on current in this particular patch clamp experiment. And so given the sort of challenges associated with transgene complementation, given this putative mechanism, we asked the question of whether precision genome editing might be able to directly address the root cause of AHC by reverting some of these mutations and potentially overcome these cellular mechanisms, providing a proof of concept permanent intervention for this debilitating neurological condition. And so among precision genome editing technologies, of course, CRISPR-Cas9 nucleases are an immensely powerful approach for precisely introducing gene disruption, as we heard this morning in the case of Casgevy. The Liu group has also pioneered sort of more precise technologies, such as base editors, which by fusion of a deaminase allow us to very precisely install transition mutations in the genome, as well as prime editors, which by fusion of reverse transcriptase allow for what's effectively search and replace genome editing. and through a prime editing guide RNA, which specifies both the target locus as well as in a 3' extension, the replacement nucleotide sequence. And so we identified base and prime editing as a potential strategy to directly correct pathogenic alleles without requiring double-stranded breaks that might lead to undesirable outcomes. And so from the outset, we recognized that particularly for prime editing, there would be a number of challenges that we would need to address. The first is that while ex vivo prime editing is underway in the clinic, in vivo prime editing, particularly in the central nervous system, does remain challenging. Second, ATP1A3 actually has a critical role in prenatal cortical development, and so it was unclear from the outset whether we could actually intervene postnatally. Third, in order to address a majority of AHC genotypes, we would need to develop scalable and parallel approaches to target multiple alleles in parallel. And finally, and perhaps most notably, there had been, prior to this work, no previous reports of prime editing rescuing a behavioral phenotype in a neurological disorder in an animal. And so a really gratifying and exciting component of this work for me as a lab scientist was getting to interact with some of these patients and community leaders who are on the front lines. And so I really am gratified to work with them, and you can't ask for more motivated people to work with. And so to answer this question, we teamed up with a number of scientists across a series of institutions. I have to highlight Alex Souza, my co-lead at the Broad, as well as Marcus Terry at the Jackson Labs, and here are fantastic patient collaborators, Natalia, Nina, and Simon. And so we set out to target in this work a majority of AHC mutations, collectively targeting 65% of AHC cases. To correct these mutations first in human cells, we thought that in principle we could apply base editing to correct 3 or 4 of these mutations. But actually, as it turns out, proximal bystander nucleotides that would undergo competitive deamination actually prevented base editing from being applied in these cases. But we were able to achieve, for a well-isolated adenine, efficient reversion to the wild-type sequence. For the remaining mutations, we turned to prime editing. And from the outset, we included benign silent mutations, shown in green. That was because previous work had shown only reverting the pathogenic mutation would result in heteroduplex intermediates that would be efficient substrates for cellular mismatch repair. And so by incorporating benign and co-installing silent mutations, we might be able to bias repair pathways toward productive editing. I'm showing just a brief subset of the optimization experiments that we performed to correct these mutations. But suffice it to say that after a substantial amount of work, we were able to efficiently correct 5 of these mutations across a number of different strategies in our cellular models of these mutations. To actually translate these conditions into a more disease-relevant cell type, we collaborated with Christine and Al at Northwestern, who had access to patient iPSCs. When we initially tried to translate our optimized correction, for example, for this particular mutation, we only saw 7% correction in iPSCs. And so this is starting from a genetic baseline of 50%. These patients are heterozygous. And so we're trying to make this bar go to 100. However, by incorporating sort of now routine enhancements to the prime editor system, including more active prime editor variants, engineered PEG RNAs that enhance cellular stability, as well as including an additional guide RNA that through proximal binding here putatively increases the chromatin accessibility of the target site. We're able to restore editing up to 70% in patient cells. Applying these strategies across the remaining mutations, again, we can efficiently, now in patient-derived iPSCs, efficiently correct these mutations back to wild type. We also assess the precision of our editing strategy applying a genome-wide nomination pipeline called Perturb-seq to assess and nominate 450 sites genome-wide. Actually looking at the editing there, we observe no off-target editing of anticipated biological significance. So in this in vitro portion of the work, I hope I've been able to show you how by using precision genome editing, we can make these cell lines, something I did not discuss today, actually optimize and refine correction strategies for these mutations, first in our HEK cell models, before finally advancing them into more advanced cell types. Moving forward, we're really excited about further scaling this technology through machine learning models, which allow us to nominate pegRNAs for consideration and actually evaluating them experimentally using high-throughput lentiviral screens. And we hope that these approaches or other more recent allele-agnostic technologies might allow us to address a broader range of these mutations. All right, so with correction of these mutations in human cells in hand, we next ask the question of whether we might be able to intervene in disease biology. And so in the absence of humanized animal models, we considered the orthologous mouse gene. And while this is over 99% identical at the protein level, the coding sequence actually differs by about 10%. And so we actually re-optimized prime editing strategies to correct these mouse mutations with the enhancements discussed previously. And we can achieve correction in heterozygous mouse primary fibroblasts derived from the animal models with those mutations. And so this is the sort of gory details of our optimized prime editing system, but suffice it to say that we can then package those into a dual AAV9 vector system, wherein the prime editor is split across two vector halves, N and C terminal, that will, upon successful co-transduction, reconstitute by protein transsplicing. And so along with a GFP marker for biodistribution, we deliver these into these animal models by intracerebroventricular administration at P0 and subject these animals to a barrage of phenotypic assays, including at the molecular, behavioral, and biometric levels. These experiments were done in collaboration with scientists at the Jackson Labs, specifically Marcus Terry, Laura Anderson, and Kat Lutz at the Rare Disease Translational Center. And so when we first assess editing at the genomic level, here's the heterozygous baseline of untreated animals. And upon prime editing, we can achieve up to 50% pathogenic allele correction in bulk cortex samples. But actually looking at GFP-positive nuclei, so these are cells that are very highly transduced, we can observe up to 90% or higher pathogenic allele correction in certain brain regions, suggesting that in some cases, we're more limited by vector transduction than by the prime editing system itself. We also assessed, in addition to genomic correction, transcriptional rescue of ATP1A3 in bulk. And this enrichment that we observe relative to DNA is consistent with the fact that AAV9 is expected to selectively target these neurons where ATP1A3 is being expressed, as opposed to, for example, glial cells. In addition to DNA and RNA, we collaborated with Kathy Swedner and Elena at Mass General Brigham to assess the functional rescue of ATPase activity in our treated animals. And relative to HC mice, which display an enzymatic defect, we can significantly restore enzymatic activity back toward wild-type levels. In addition to molecular readouts, we also assessed a number of critical behavioral phenotypes, most notably this hypothermia induction assay. So here, mice are exposed to a cold water bath to induce episodes of dystonia, hemiplegia, and other seizure-like events that's typically observed in patients. And so mice are assigned a qualitative score ranging from normal behavior all the way to severe cases where the mouse is unable to right itself. We also quantify the number of seizure-like events and measure the amount of time it takes for these mice to first regain writing and then subsequent normal voluntary movement. And again, patients also experience these types of symptoms, and so in response to harsh stimuli and those types of triggers. So relative to HC mice, which score very highly on the scale, we can significantly reduce the qualitative severity of these episodes. But more excitingly and quantitatively, we can measure the most severe events, specifically these tonic-clonic-like seizures. And relative to HC mice, we can essentially restore the number of those events back down to baseline. Measuring the amount of recovery time for writing and also subsequent mobility, we see that these HC mice have a sort of shifted recovery time where they take much longer to recover from these hypothermic events, and prime editor treatment shifts that back toward wild type. Beyond these types of induced phenotypes, we also measured coordination using the rotarod assay, and we can restore coordination in prime editor treated animals up to or almost back to wild type levels. Most notably, HC mice, like some patients, can display a survival deficit due to spontaneous episodes, and HC mice do not survive out to the study endpoint of 52 weeks. By contrast, prime editor treated animals, we can significantly enhance survival and reduce instances of spontaneous death. So I hope I've been able to show you across the central dogma and also at the level of behavioral and survival readouts, we've been able to intervene in HC disease biology. A sort of key missing link that we're excited about exploring further is using in vivo electrophysiology to actually assess to what extent prime editing might be able to intervene and partially rescue this type of circuit or higher level complexity. And this is in collaboration with Arne, Georgie, and Elsa at Leiden University. And sort of going to the ladders to cures idea of identifying notable biological mechanisms, I think in the case of therapeutic genome editing, maybe the biological hypothesis is often quite simple. But I think now that we have these tools, we can actually use them to study biological questions related to disease and its progression. For example, at what level does correction at the protein level translate into organ or behavioral level phenotypes? As we advance toward the clinic, we're also thinking about a number of other studies related to dosing, more relevant routes of administration in the clinic, as well as later ages of intervention. And so that's all I'll share today. But I hope, moving forward, that we'll be able to accelerate progress through the approaches mentioned previously, and that this work will inspire the creation of humanized models or allele-agnostic methods that will allow us to target an even broader range of these mutations moving forward. And so with that, I have to thank David, of course. I mentioned co-leads Alex Souza and Marcus Terry, these sources for funding, and I'll be happy to take any questions during the Q&A.
Well, thank you, Holt. That was a great talk and very, very exciting data. As with the previous sessions, we're going to do questions after the next speaker. We'll do it all together. So let me introduce you the next speaker. It's a pleasure to introduce Dr. Rajat Gupta. Raj Gupta is an assistant professor of medicine at Harvard Medical School with a research lab in the Division of Cardiovascular Medicine and Genetics at Brigham and Women's Hospital, and also here at the Broad Institute. And as a physician scientist, he is interested in how genetic mutations affect vascular function, especially to find new treatment targets. And that's what he's going to talk about today in the talk titled Toward a Cure for Cerebral Cavernous Malformations, Genetic Insights and Perturb-seq in Action. I'm very excited about the in action part.
Right, yeah.
Welcome.
Yeah, thanks. Thanks so much for that introduction. Is my mic on? Can you hear me? Yeah. Okay, perfect. Well, wonderful. So I did my postdoc here at the Broad Institute many moons ago with David Altshuler, and the goal was to identify the genetic drivers of common diseases like coronary artery disease and hypertension. And that's what I started my lab to study. but by sort of curiosity-driven research, as Anna likes to say a lot, we identified that there were these nodal pathways shared between common vascular diseases and rare vascular diseases. And so this community of Ladders to Cure has sort of helped us hone in on, is there some therapeutic potential for insights made from common vascular diseases on a rare disease? And that's the story I'm going to share today. Okay, so the disease that I'm going to talk to everyone about is cerebral cavernous malformations. There's been a big focus on neurodegenerative disease, neuromuscular disease, and this is a neurovascular disease. And so it's extremely rare. There's a Mendelian form, which is 1 in 5,000 people, and then there's a sporadic form, which is 1 in 200 people, so more common. The Mendelian form is driven by 3 mutations, all named for the disease they cause: CCM1, CCM2, and CCM3. And what you see in these pictures, it's almost like a blood blister of the brain. It's like an enlargement of the venous structures that results in these enlarged blood vessels that can burst at any time. On the right, you see an MRI, and each one of those black dots shouldn't be there. They can burst unpredictably and result in hemiplegia, stroke, and death. And the only real proven therapy is neurosurgery, and it's a very painstaking neurosurgery. Only the sort of most skilled neurosurgeons, the chief of neurosurgery at Children's Hospital here, is one of the world leading experts. And they have to sort of individually excise each of these lesions. And they don't know which lesions are going to burst. Many of the lesions form in inoperable regions of the brain, like the brainstem, which they can't go after surgically. And there's no therapies that delay or reverse the disease. And so, I got super interested in this disease because of our Perturb-seq work that I'm going to share with you. And so I asked every neurologist in the city to send me their CCM patients. And somewhat embarrassingly, every time a patient comes sees me, they say, "So you're a neurosurgeon, right?" And I sort of have to say, "Well, no, I'm actually a cardiologist. I'm just really interested in this disease. I think we could cure it one day." And so, some of my patients will even say, "But you're still a neurosurgeon, right?" Because they believe that that's really the only way to treat the disease. But the exciting part that I like to explain to the patients that I talk to is that their rare disease is teaching us about the common diseases of the blood vessels and the other way around, right? And the reason for this is the common diseases that we study, right, like coronary disease and hypertension, they're driven by these small effect size variants where we don't know the causal genes. We often are assuming that we know the causal genes but are often wrong. Whereas rare diseases like cerebral cavernous malformations or thoracic aortic aneurysms are driven by rare variants identified in familial studies and are linked to severe phenotypes, but they both converge on the same nodal pathways. And in endothelial cells, that's been increasingly apparent from our Perturb-seq work. And what I'm excited about, and it's something that I think is we haven't talked about it today, but this sort of virtuous cycle between rare and common disease, right? When we did Perturb-seq, we only knew the biology of the genes we were identifying because of prior work in rare disease. And I think the rare disease work is informing the common disease work, and hopefully the other way around. And so the outline of what I'll present today are that, from Perturb-seq, we have identified central nodes for vascular disease risk, and the node that I'm going to talk about is the regulation of KLF2 and 4, transcription factors that are very involved in shear stress response, the response of endothelial cells to blood flow. But there's this opposite direction of effect between CCM and CAD genetics, and so what are the insights we can make in that? So finally, the work that was funded by this Ladders to Cure seed project was to define the causal pathway for CCM better. And that was to look for cellular phenotypes with cell painting, work that J.T. Neal beautifully presented today. And then finally, we've sort of begun the effort to develop therapies in this space. And so the work that sort of motivated our entry into CCM was an endothelial cell Perturb-seq screen that was done by Gavin Schnitzer and Helen Kang in our lab in close collaboration with Jesse Engreitz, who's now at Stanford. And we were trying to study the 228 non-lipid-associated CAD genes that were identified from GWAS studies. We wondered if we could study all of the possible causal genes in endothelial cells at the same time. And so the project that we embarked on was perturbing every possible causal gene. So in a pooled format, use CRISPR-I to knock down expression of each one of the possible causal genes within a megabase of each of the GWAS variants, and then perform single-cell RNA sequencing to look for the transcriptional effects of each of these perturbations. So in the middle panel, you'll see that we were able to successfully sequence 215,000 single cells with 37,000 different guide RNAs and 90 cells per target gene. And then we used methods that were developed in Pardis Sabeti's lab here to look for the co-regulated gene networks from this analysis. So you find, like de novo, which genes have effects on which genetic programs in vitro. And here's like sort of what we call the wiring diagram of the endothelial cell that's sort of de novo analyzed from our Perturb-seq data, right? No assumptions are made. These are just the genes that travel together, in our experiment. And you'll see that there's 50 co-regulated gene programs, 37 of which you would have found in any cell type. They affect like common processes like cell cycle or ribosomal biology. But 13 of the programs are endothelial cell specific, and they are in the sort of pathways that you would expect to be associated with vascular diseases. And the one of these programs, topic 8 or program 8 from our analysis, was enriched for CAD genetic risk. So 41 of the 241 CAD risk variants all converged on this pathway. And the pathway is sort of shown here in this cartoon. Genes that were upstream of KLF2 and 4, which is sort of the nodal piece of the pathway, they're in a pathway called the CCM pathway. And that's sort of depicted in the middle of the diagram. But genes upstream and downstream of KLF2 sort of unite to show genetic enrichment for all of these variants. And so one of these genes is CCM2 itself. And we had never heard of CCM2. We'd never sort of heard about the disease itself. But what we were seeing is that loss of function of CCM2 and other genes in that complex were protective for coronary artery disease. So now we sort of, after reading the literature, we had this sort of dilemma. The literature is telling us that large, rare deletions in CCM2 are causing this severe neurovascular disorder. But then when you look at a common missense mutation in that same gene, it's protective for coronary disease. And so our Ladders to Cure project was to really confirm these opposite directionalities, really see if that's true, and to better define these phenotypes. And so to begin with, why is there opposite directionality? So on the right you see work done by Mark Kahn's lab at the University of Pennsylvania, where they made the CCM knockout mouse model. And they made it for a CRIT1 knockout, which is also CCM1, which you see in the top right. And then in the bottom left is the CCM2 knockout. You see these brain lesions that are forming in the cerebellum. When they knock out MEC3, which is just downstream of the CCM pathway, it's the sort of kinase cascade regulated by this complex, they rescue it. So they cure it in mice by knockout of MEC3. So they say that the pathogenesis of the brain disease is high KLF2. And we also think that's the protective effect in CAD. So why could this be? Why are there these opposite directionalities? It could be that there's specific biology at that missense mutation. It could be that there's a difference between complete and partial loss of function, sort of a dosage effect. It could be because arterial and venous ECs respond differently to mutations. And it could be because there's a difference between developmental effects and adult effects. These are the kind of questions that will probably take my lab the next 10 years to study. But I think they're really important and exciting. And so just to begin with, what have we done like with the Broad community and the Ladders to Cure community is that we found that the coding mutation that's associated with decreased CAD risk is at this one spot in the CCM protein. And its minor allele frequency is 8%, so it probably doesn't affect risk of a rare disease. But the CCM causing mutations are all sort of on the C-terminus end. They affect maybe other properties. And so what are those other properties? Are there different proteins that bind at each site? And one of the things that binds on that C-terminus end is a new protein that had never been linked to this complex. It's in orange here. It's called T1RD1. So our hypothesis was that different cellular mechanisms may be responsible for CAD or CCM. And so to sort of unlock this, we use the cell painting or variant editing approach that JT Neal nicely introduced, right? They use machine learning of microscopy images to identify the effects of different mutations. And they have standard cell painting and then variant cell painting, which you heard about earlier today. So we kind of ran through a lot of mutant alleles in CCM. And what you see is the reference allele for CCM at the bottom, the wild type allele, results in cytoplasmic localization of CCM, right? All the green, all the GFP tag CCM is all in the cytoplasm. But a specific variant allele at the C-terminus end of the protein, this isoleucine 432 to threonine, changes the localization completely. It's sort of one of the examples JT talked about, that now it's more nuclear localization. A different amino acid change at that same residue has no effect. It's still cytoplasmically located. And so, now we want to expand that. So we are finding specific phenotypes for disease-associated mutations for the rare disease that aren't seen with the common disease. And now we want to see if all the genes in this pathway have similar morphologic effects. So Jia Hao, a new postdoc in my lab, has sort of run this analysis. And this is work we did with the imaging platform here to knock out all of the sort of topic 8 genes that were enriched for CAD and all of the CCM genes. And they don't all cluster together. In Perturb-seq, they all show the same transcriptional phenotype, but morphologically, they cluster separately. And so finally, I want to just spend the last few minutes talking about, is there an opportunity for therapies that target what we've learned? And so we want to make the case that we could repurpose a drug that was actually developed by Nat Gray when he was here at the Broad and Dana-Farber. It's an ERK5 inhibitor. It's part of this pathway. And so the cartoon here are all the sort of functions of the CCM complex. You see CCM1, CCM2, and CCM3. They bind each other. They bind MEC3. They affect KLF2 and 4. Our data suggests that's the mechanism of disease. But the current therapies in development are targeting peripheral mechanisms. There's a clinical trial in progress to use statins, which target HMG-CoA reductase, sort of not a central member of this complex. Recursion, with work that was actually done here at the Broad with Ann Carpenter, is using Tempol, which targets reactive oxygen species. And then other companies are making ROCK inhibitors for this disease. And like I said, those are sort of in that dotted circle. They affect stress fiber formation. That's an important mechanism of disease, but we don't think that's the central mechanism. That's not the node that keeps showing up in all of our experiments. The node is really the genes that are driving disease risk. And so if we wanted to target that, we found in the literature that Nat Gray, a great chemist who had made PROTAC molecules for ERK1 and ERK2 for cancer, had made an ERK5 PROTAC. And Nat Gray's so famous that even if he shows his compound does nothing, he can publish it in Cell Chemical Biology, which he did very nicely, right? So this is they made the compound. It was extremely specific, as you see in the bottom right, RNA-seq, and it was a very potent inhibitor of ERK5 and downstream signaling, but it had no effect on cancer. So then they stopped working on it. But we think that we have knockout cells, CCM2 and T1RD1 knockout cells, that are resistant to shear stress. They demonstrate some of the phenotypes that you'd expect. And so at the bottom, kind of the most striking phenotype is that the CCM2 and T1RD1 cells have stronger stress fibers. And right above that, you see that even with flow, the wild type cells will all align, but the knockout cells sort of retain this disorganized pattern. And so we tested their ERK5 PROTAC. And there is a therapeutic range where you could take the CKO or CCM2 knockout cells, and they will normalize KLF2 expression, whereas wild type cells will have a minimal response to KLF2 expression. So we found this to sort of be a possible targeted therapy to the central mechanism that might be promising for CCM, and we'd like to expand that work. And so I'll end by saying we're just kind of scratching the surface of how nodal biology could affect one rare disease and multiple common diseases. But to go forward, we want to develop in vivo Perturb-seq methods to identify the co-regulated networks in a disease context. And we're starting along those lines, with AAVs that target endothelial cells that were actually made by the Deverman lab here. We've been able to get pretty good expression in a CRISPR-I mouse. We want to determine the different in vivo effects of CAD and CCM-associated mutations. We want to test the efficacy of this first compound for possible lesion resolution in mice that get CCM disease. And then finally, I didn't have time to talk about this, but we've partnered with the foundation. So there's an alliance to cure cavernous malformations that kind of enrolls all the patients who have this disease. And we want to, now that we have Perturb-seq data, we could take all the genotype negative families and see if they have mutations in related genes to this pathway. And so with that, I'll thank the people who did the work. Gavin Schnitzer, Helen Kang led the Perturb-seq, and multiple members of our lab. Jesse Angreitz's lab has been really instrumental in almost all the work. And we always thank the families that have contributed samples and their time to this research. Thanks.
Thank you so much for this talk. I want the two speakers maybe to join me up here, and we're starting our Q&A session. Just go and grab a microphone if there's any questions. If there's none for now, I'll start. Holt, I have a question for you. You kind of in the beginning kind of presented that, you know, you have a disease, you have a gene, you have the mutations, and then you tried with base editing, but there were off-target effect, and then one maybe works, the other one didn't, and then you went to prime editing. Do you think that will kind of be the pathway forward for, you know, if I have a gene of interest, and that would be kind of the pathway that that everybody has to go through?
Yeah, I would say, and this happens quite often in our lab, where people will, you know, see a mutation and at least try base editing. It's possible that, you know, even if it's like 2 nucleotides away, if the window is just right, you can get just on target without any bystander. And that sometimes surprises us too. Like sometimes we'll make calls like, oh, there's no way base editing will work, but let's just try it anyway, and surprisingly it does. But yeah, I would say given the additional maybe experimental requirements and throughput for prime editing, we usually put that as a second choice.
But let's say prime editing will get more and more efficient. Would that maybe overtake the base editing?
Yeah, that's an interesting question, and we certainly hope it will get there. I think now we're starting to see where our most optimized prime edits can start to hit the levels of like some of the earlier base editors. The base editors are still just like monsters at deaminating DNA. And actually for those, off targets become much more of a problem. So prime editing is much better due to sort of the 3 successive DNA hybridization events that have to happen in the mechanism for off target limitations, yeah.
I had a question for Raj. I was wondering if whether in the CCM patients or the mice, if you studied the arterial endothelial cells, what phenotypes would you see?
Yeah, that's a great question. So we're studying that in the lab because that's like sort of the best evidence that there's a different protective effect. And the short answer is we see a protective effect in the CCM heterozygous mice. Homozygous CCM2 knockout mice don't survive in germline. Inducible CCM2 knockout mice exist and they can survive. We haven't studied them, but we thought a heterozygous mice mimic the effect of like a modest germline common mutation. And Shi Fang in my lab is got really nice data that they get less athro and they get less vascular inflammation. So you do see that loss of function, modest loss of function is having a protective effect.
I'm Ashley Conard at Microsoft in the Broad. I really appreciated both of your talks. I had a question about the relationship that you drew between identifying mechanisms that allow you to draw parallels between rare and common disease. Mm-hmm. Have you thought about ways or have you identified any patterns that will allow us to help making those relationships as we think about therapeutic strategies that might allow us to kind of shift between applications in rare and common disease space?
Yeah, that's a great question, and Anna has like great experience with this as well, and I think we're all trying to do this to some extent. Our approach was like cellular genetics, so Perturb-seq, identify pathways and nodes, using perturbation in a relevant cell type, and then you'll see this convergence of genetic risk on certain pathways, and then the pathways are probably already described from rare disease. So that's like a more laborious way of doing things. I think it requires this sort of high throughput cellular phenotyping. But we're just talking about transcriptional pathways there. What about like morphologic pathways? Well, maybe like variant cell editing and microscopy could do that. And then I think you could harness the pleiotropy of variants, right? So as more sequencing becomes available, maybe you could just find it from like lots of sequencing of biobanks. UK Biobank's not really powered yet for something like CCM, but we would expect that there should be like convergence of certain mutations on like hemorrhagic stroke and CAD risk. But it's just not powered yet with only 500,000, but maybe at like 1 million, 2 million, it would be.
Thank you.
So I had a question about the efficacy between the Hexel models that you were using and the mouse models. And I thought it was super exciting that the efficacy was so high in the mouse models. And I was wondering, what challenges there are like going forward with efficacy like on the mouse model side compared to in vitro.
Yeah, that's a good question. It's always a little bit disappointing when we do all this work in the Hexel and then it just doesn't translate always. Fortunately, we do have some approaches now that allow us to elevate efficiency in more primary types. But yeah, your question about, I guess, translating from in vitro to in vivo editing is a hard one, and I would say that for us, we typically try to assess in the most advanced system possible and as early as possible, just to get a sense of what those challenges might be, and whether I guess there are alternative editing strategies that you might want to pursue or prioritize. Yeah.
So regarding the prime editing in vivo introduction of indels, even though it's a fairly low number, is this something that you're concerned with? And is there anything in the mechanism of how they get introduced that you could try to leverage as a way to minimize them?
Yeah, that's a good question. So I think, so for one of the mouse models, we had, I don't want to go too much into the detail about the specific complexities of the prime editing system, but one of the mouse models had what we call like a PE3B nicking. So we have a second guide RNA that nicks the opposite strand to bias DNA repair. And in some cases, that nick can be selectively introduced only after the edit has been introduced. And so when we use that type of strategy, we typically see very low indels in vivo. But for the other mouse model where we didn't have that requirement that the mutation already be corrected followed by nicking, and it just nicked elsewhere, we actually observed much higher indels. So something that we're following up on that to reduce the number, that percentage, is to modify the strategy to have that second nick be concurrent. I will say that I guess it will certainly depend on your particular disease of interest, whether I guess indel-mediated, say, nonsense-mediated decay might be relevant for your particular protein. But at least I think as long as it's leading to non-functional variants that are just, I guess, truncated, that don't compete, then that might be okay, depending on the disease as well.
Okay, well, I wanted to thank the speakers for 2 excellent talks and for all the work they do. Thanks for the discussion, and we can move on.